Hello everyone, welcome to my channel. First of all, I am thankful to all of you that you watched the previously uploaded video in which I gave the solution of section A, B, C of first set of 57/3/1 series. Okay. Section D consists of two questions which are case based. Question number 29. In recombinant DNA technology, restriction enzymes are used as they recognize and cut DNA within a specific recognition sequence. BAM-H1 is one such restriction enzyme which binds at the recognition sequence 5' GGA-TCC3' and cleaves this sequence between G and G, here. On each strand, whereas ALU1 binds at the recognition sequence 5' AGCT3' and cleaves these sequences between G and C, it means here, on each strand. Okay, so now you have already understood that when the restriction enzyme cleaves two bases at the ends, away from the center, the sticky ends are formed. So here blunt ends are formed because the restriction enzyme work at the center. It is a sequence of 4 bases and ALU1 works in the center at GC. So blunt ends are formed. This is away from the center. That means away from AT sequence, it cleaves the bases between G and G. So now here sticky ends are formed. Question is, if ALU1 is used to cut the given DNA strand, how many DNA fragments would be formed? Write the sequence of each fragment formed with its polarity. So let us see which sequence will be there. GGA-TCC. GGA-TCC is not there. AGCT, it's here, and AGCT again here. It means when ALU1 type of restriction enzyme works on this DNA strand, there will be 3 DNA strands formed. One is this CCGTAG, 5', 3'. Another is CTA TCAG, 5', 3'. CTA TCAG, and the third one is 5', 3', CTGG. So these are the 3 DNA strands which will be formed by ALU1. Now write the sequence of each fragment. We have already written the sequence 5', 3', 5', 3', 5', 3'. Next is, which one of the two restriction enzymes, BAM-H1 and or ALU1, will preferably be used on the same given DNA strand to make a recombinant DNA molecule, and why? Obviously, ALU1 will work on it because there is no sequence on which BAM or BAM-H1 works. So ALU1 will work on it. Now you can say that by ALU1, sticky ends will not be formed, but blunt ends will be formed, and blunt ends can be ligated by DNA ligase, though it is less efficient, but ALU1 can be used to cut the DNA. After binding to the 2 strands of the double helix DNA, where specifically does the restriction enzyme act to cut the 2 strands of DNA? The answer is restriction site. Write the specific term used for the specific nucleotide sequence of DNA recognized by a restriction endonuclease, that is a palindromic sequence. Palindromic sequence. A choice is there. Write the specific sequence of DNA segment recognized by the restriction endonuclease EcoR1. Very simple. G A A T T C. 5 prime to 3 prime. Question number 30. Study the figures given below that depict the comparative age distribution of human populations in Sweden and Rwanda. International database is given of 2003 year, and answer the questions that follow. As you can see in this diagram, percentage of population and age is given. This data is for pre-reproductive age. 10 to 14 years, 5 to 9, 0 to 4. 2.7%. It means the reproductive population will be decreased in the future. Number of males and females in the future will be lesser than the present condition. Understood? So, this is a declining type of population pyramid or age pyramid. Another one. For Rwanda, in this age pyramid you can see that the percentage of population males and females 0 to 4 year, 5 to 9 year, 10 to 14 year, 15 to 19. Earlier it was for 20 to 24 years it is 4.6%, whereas in the reproductive case it is more. It means in the future the reproductive population will be more. The number of males and females of reproductive age will be more. So you can say that in the near future the population will increase. Okay. Now let us see the questions. The A part here is what can be inferred from the very broad base of Rwanda's age pyramid? Support your answer with the data provided in the figure. Reason for this is that the males and females in the pre-reproductive category is more than the present condition of reproductive age. So this is the reason. Second question, Sweden has an age distribution that is approximately of the same width near its base as at the apex. Here, this is the pyramid of Sweden. So this will be of declining type. Declining type of age pyramid is this because the population which fall in this category of the reproductive phase is lesser. From 3.1% it has been declined to 3.6%, then 2.9, 2.7. So in the near future the population of Sweden will be decreased, will be lesser. C part, name the type of age pyramid shown above the Sweden. Now you know that for Sweden it is declining type and for Rwanda it is expanding type. Next question. So we have finished here section D. Section E. Explain any 4 devices that flowering plants have developed to encourage the cross-pollination. To encourage cross-pollination, plants develop some outbreeding devices. And these are, number 1, when there is absence of synchronization between the pollen release and stigma maturity. Stigma reception. So, no synchronization between pollen release and stigma reception. This is the first point. Second is self-incompatibility. Self-incompatibility. It means when the stigma rejects its own pollens. Stigma of the flower rejects the pollens of its same flower. Third is production of unisexual flowers. Production of unisexual flowers, like you have seen in monoecious and dioecious plants, there are unisexual flowers produced, male and female flowers are produced. Fourth is the anther and stigma are present at different height. Different height. You can also write if there is a barrier between the anthers and the stigma, like hercogamy. Why do plants discourage self-pollination? Simple, to prevent inbreeding depression. So, to prevent inbreeding depression, the plants discourage self-pollination. State any one reason. Why do plants? To prevent inbreeding depression, which results in loss of vigor, or you can also write loss of variations. Fine. Now the B part of this 31st question. Explain the ovarian and uterine events taking place along with the role of pituitary and ovarian hormones during menstrual cycle in a normal human female under the follicular phases. See what is to be written here. Explain the ovarian and uterine event. ओवरी में कौन से इवेंट्स होते हैं, यूट्रस में कौन से इवेंट्स होते हैं, साथ ही साथ रोल ऑफ पिट्यूटरी एंड ओवेरियन हार्मोन, ये सब लिखना है आपको. तो ओवेरियन में क्या लिखना था, ओवेरियन में एट दिस फॉलिकुलर फेज ऑफ प्रोलिफरेटिव फेज आपको लिखना था कि एट दिस टाइम द फॉलिकल डेवलपमेंट takes place which results in the maturity of Graafian follicle. तो follicular development होता है और mature Graafian follicle बनता है. किसमें? ओवरी में. यूट्रस में क्या होता है उस समय? At that time the endometrium lining of the uterus regenerates. तो यूट्रस में आप लिखेंगे endometrium lining regenerates. Now, role of pituitary and ovarian hormones during the follicular phase. The role of pituitary hormone कौन से होते हैं? FSH और LH. So, their secretion increases to develop the follicles. And ovarian hormone, only the estrogen is secreted at that time. So, the level of estrogen increases. Next is luteal phase or secretory phase. Now for the luteal phase or secretory phase, ovarian event and uterine event. Ovarian event में rupture of Graafian follicle or ovulation takes place. For uterus, endometrium is maintained. About the pituitary hormone, LH is secreted at its maximum level. And for ovarian hormone, now graphene follicle is ruptured, it means corpus luteum is also formed. You can write here corpus luteum formed. And ovarian hormone, progesterone is secreted. By corpus luteum. So this way you had to write the answer. And the last one is menstrual phase. So for menstrual phase, same ovarian event, uterine event, role of pituitary hormone, ovarian hormone. Now first is ovarian. Ovarian event, what happens? Degeneration of corpus luteum. Uterine event, the lining of endometrium breaks down. And what about the role of pituitary and ovarian hormone? So FSH and LH and ovarian hormone also, progesterone and estrogen hormones, these are secreted at the lowest level. Their level is low in the body. Next question 32. The influence of both the alleles in a heterozygous state is clearly expressed in codominance. Explained with the help of inheritance of ABO blood group in humans. You know that ABO blood group is determined by a gene called I gene. It is determined by I gene, and I gene has three forms of this. Three alleles of I gene are there: IA, IB, I naught. And when we make different combinations, we will find IA homozygous condition, IA, IB heterozygous condition, IA, I naught again heterozygous condition, IB homozygous condition, IB, IA that we have already written here, and IB, I naught, and the last one is I naught. Only these 6 combinations can be there. IA will result in A blood group, IB in AB. Codominance is shown here. It means in case of IA, I naught, A is dominant. In case of IB, I naught, B is dominant. But when the heterozygous condition IA and IB, both are dominant. Okay, and both of these alleles express themselves. It means both type of antigens are formed on the surface of RBC, A and B type. Here A type, B type, here again B type, and this one is the recessive condition. Which blood group? O blood group. So the influence of both the alleles, you had to show this. When IA-IB condition is there, AB blood group is appeared. I hope I am clear. So B part, a group of genes are regulated and expressed together as a unit in leg operon. Explain the mechanism of switching on of the structural genes of leg operon. Leg operon is a polycistronic structural gene with which is regulated by a common promoter and operator gene. So let us see the diagram of this. Promoter, then this is the regulatory gene, then again promoter, operator, then one of the gene Z, another structural gene Y, and this one is the A structural gene. I have written here common promoter and operator. So O for operator and these are the promoter genes. I is actually a regulatory gene. You have to write all this. Regulatory gene. This is operator. So what happens? This I gene is actually responsible for the formation of a repressor mRNA. Repressor mRNA. And this repressor mRNA, when it is translated, a protein is formed which is known as repressor protein. In the absence of an inducer called lactose, what happens when inducer is present in the medium, lactose is present in the medium, this lactose inactivates this repressor and prevents this repressor to bind on this operator. So this way the operator is switched on and the process of transcription takes place. Here this LEC mRNA is formed, LEC mRNA, and then from different structural genes, different enzymes are formed. Like from this beta galactosidase is formed. From Y gene, permease is formed, and from A gene, trans-acetylase is formed. So this way we can say that the lactose prevents the repressor protein to bind with the operator, and this way this operator is switched on and the process of transcription and translation takes place. Next part, regulation of LEC operon is referred to be negatively regulated. Justify giving a reason. We have just discussed the switching on process of the operator. Regulation of LEC operon is referred to be negatively regulated. You know, from the I gene, repressor protein is formed all the time. Repressor protein is formed all the time. Okay, but when the lactose is not there in the medium, in absence of lactose, what happens? This repressor protein binds with the operator and switch off the process of translation. So it prevents RNA polymerase to transcribe LEC mRNA. So this way we can say that it is negatively regulated, because at the time, in the absence of lactose, what happens? The repressor protein switch off the operator. Question number 33, the last question of this question paper. Describe the life cycle of Plasmodium from the time it enters the human body till a female Anopheles mosquito bites an infected person. For this you have to draw a part of the life cycle of Plasmodium. Plasmodium enters into the human body in the form of sporozoites by biting of female anopheles. Then what happens? These sporozoites infect the liver. They reach the liver through the blood, infect liver cells by reaching through blood. Inside the liver, these sporozoites multiply with the help of asexual reproduction. Sporozoites multiply by asexual reproduction burst the liver cells and infect RBC. Then what happens inside the RBC? In RBC, these cells of Plasmodium further multiply and asexual reproduction takes place. Burst the RBC by causing cycles of fever and other symptoms like chill, by secreting hemozoin. Then what happens in RBC? Male and female gametocytes are developed. Gametocytes are developed. Now you can see that what happens when another female anopheles bite. The infected person, what will happen? These gametocytes will enter into the female anopheles. And when this female anopheles will bite another healthy person, the other form of Plasmodium will enter into the healthy person. Second part, mention the two events of Plasmodium life cycle that occur within the female anopheles body. You have just written that when the female anopheles bite the infected person, what happens? The gametocytes enter into the mosquito, and these there, it means fertilization of gametocytes takes place in the gut of mosquito And second point is these fertilized gametocyte escape from gut and enter into the salivary gland. Into the salivary glands of mosquito. Enter into the salivary gland. Next is, there's a choice. Write two differences between malignant tumor and benign tumor. Very simple. Malignant tumor and benign tumor. The cells. So you had to write these two differences between the malignant tumor and benign tumor. The neoplastic cells or malignant tumor cells grow very rapidly, invade and damage the surrounding tissue. And the type of these type of cells remain confined to their original location and do not damage the distant tissues or organs. Whereas for malignant tumor, the cells show property of metastasis and cause new tumor at distant places. Benign tumor cells do not show property of metastasis and cause little damage to the body. So this is the answer of first part of the choice question. And another is, explain any three diagnostic techniques for the detection of cancer. Very simple. You know that surgery, chemotherapy, radiotherapy, immunotherapy, these are different categories of diagnostic techniques. And you can also write immunotherapy, by using beta interferons or alpha interferons. Thanks for watching.